Good morning.
Good morning, sir.
Good morning, sir.
Yes. Okay. So today we have only 13 students on board. Why so less attendance? Fine. So let's begin. So maybe till talking about the cellular metabolism, where as I told you that if you want to go for any kind of change within a particular system, a particular cell, okay, means it could be a cell of any life form. It could be a prokaryotic or it could be an eukaryotic cell. The first thing what we should understand is the system itself. Okay, and to understand the system, we'll have to understand the metabolism which is taking place, because metabolism is the one which is responsible for providing the uh, carbon, okay, which is involved in the overall production of the biomass of that particular cell, which is responsible for the growth. Along with that carbon, you also require energy. Okay, now this energy, it could be in the form of ATP, it could be the form of NADH or the NADPH, which overall helps in carrying out the different type of the metabolic reactions. That is one thing, so that the biomass can be increased, the cell can grow on the different type of the activities. So it could be the maintenance, the repair mechanisms, etc., etc. So for all those particular, which could be obtained from the carbon source which has entered into the cell, okay, which was an input inside the cell. So after having all these different type of the activities, there are some products which could be secreted, or there are some products which are excreted outside the body. Right. So this is what we try to understand. Hmm? So we had focused only on one particular path, the central path, which could be common for all the life forms, whereby though that particular path is responsible for production of many different type of the building blocks, which is also responsible for the production of energy, and that is nothing but the entry of your glucose or your carbohydrate, which is undergoing the glycolysis pathway, then the TCA cycle, and then the electron transport chain. Right. So we are not considering the whole metabolic pathways. We are considering only one particular central path. Okay. So here we have tried to understand that how these metabolites, which are into the medium, how it comes inside transport mechanism, what we are seeing. So through the transport mechanism, it will come inside, it will undergo the glycolysis, the TCA, the electron transport chain. Different paths, they are occurring in the different compartments. So for that also you require the transport system. So in toto, we try to understand, okay, that what are these transport systems, the different type of the transport system, what are the different metabolic pathways which are involved into it, right? That too we have talked only about the central path. Now this pathways what we have talked about, it is not that they are continuously occurring, okay, or they are carried out in the same proportion. No. So depending upon the requirement of the cell, whether the cell want the carbon compounds, whether the cell wants the ATP, etc., etc. So that is going to decide whether this pathway is active or whether it is inactive. That is one thing. Then the intermediates which are present within this particular path, maybe your glycolysis or the TCA cycle path, these intermediates, they are not always at equilibrium, or they are not processed at the same rate. So each and every intermediate could be present in different concentration, or each and every intermediate can have the different type of the fates, because these intermediates, they are also involved into the different pathways. There could be n number of branches to the central path, whereby these intermediates could be diverted toward those particular Okay, so one example which I have told you about the glucose 6 phosphate. So this glucose 6 phosphate, if the cell wants an energy, yeah, this glucose 6 phosphate will go toward the glycolysis part. If the energy is not required, it can go for the synthesis of the glycogen. Okay, or if the cell requires an reducing power, that is an NADPH, okay, so it may undergo the pentose phosphate pathway, right? So that is what you can say about the glucose 6. So that is what you can say about the glucose 6. So the flux, okay, the flux of this glucose 6 phosphate could vary depending upon the need of the cell. If the cell requires a high amount of energy, so you can say that the flux is toward the glycolysis path. If the cell is requiring NADPH, so the flux is toward the pentose phosphate path. If any one of these is not required, that is, it has a sufficient amount of ATP, sufficient amount of NADPH, so in that case, you can say that it can be what you can say diverted toward the production of the glycogen where it can be stored. Right. So like this, there are different fates. Then you have the pyruvate, where pyruvate, if it is condition based, so if it is in oxygen condition, you can say that pyruvate is converted to acetyl CoA and oxidative phosphorylation takes place. If there is an anaerobic condition, so there is a possibility that it may be either converted into lactate, or it could be converted into the alcohol, or any other type of the fermentation process which you are aware of. So you may get the different type of the products right. So this is what we try to understand through the topic what I have explained you since last 2 days, that is called as the cellular metabolism. So that base has been created. Now we'll try to move into the forward direction, that what actually should be done when you are going for metabolic healing. So if you remember, I told you about the 4 steps, that is DBTL. Okay, so here what we have done, we have decided upon a particular system, that is the host has been decided. You might have decided for some particular metabolite, like the example what I told you about the succinate, that you want the succinate to be understand that what are the pathways which are involved into it. So we try to see that it is the central pathway. Fine. So now we'll have to go in more details. We'll have to we'll have to create our own model. We'll have to construct our model. Okay, where through that model we'll have to try to understand that what are the fluxes of each and every reactions which are taking place into that pathways. Okay, so for that we require some mathematical, we require some statistical type of the constraints, okay, or the factors which will help us to create our own model. And if we have the model in front of us, it will be easier for us to go for the different type of the modification. Okay, if you are if you want to construct a building, we first prepare an uh, model onto the paper. So initially, or in the previous days, it was done onto the tracing paper or some those kind of paper. But nowadays we have the sophisticated thing, that the laptops or the computers are there. There are many softwares are available. So simply if you put the things what you want into that program, it will give you the overall picture of that building which you want to construct. Okay, and there you can modify it, that the width, if it can be increased or decreased, or the size of the rooms, if you vary, what kind of effect it will have, etc, etc. Now I think you start imagining what I want to say here. So the same thing we are trying to apply here. Fine. So now to proceed further, first let me present the screen, and then we'll go for it. Is the screen visible?
Yes, sir.
Yes, sir.
Okay. Okay, so we have understood the system, and from that system now we'll try to design the metabolic network. So that is what I'm trying to explain you today, that what do you mean by this metabolic network, and how you can go for having that particular network. Right? So that is what we'll try to understand today. So yeah. So bioenergetics, this also we have already discussed, or in the last 2 3 lectures, what I had been talking about, it was related to the bioenergetics only. So, so living cells, as we know, that it continuously require the energy for different activities, the transport, motility, the overall maintenance, etc., etc. And the energy what we obtained, it is from the carbohydrate source, right? So bioenergetics is what you can say, a thing where already we have talked about it, but we are trying to see it in one this particular slide, that you have the substrate. So this substrate, in maybe in presence of the oxygen, carbon dioxide, water, some things, the substrate which is entering into the cell, it will be undergoing a process that is called as an catabolism. Through catabolism, what do you get? You get the different type of the precursors, the different type of the carbon compounds, what you can say here. Along with that, you get the energy in the form of either ATP or the NADPH or the NADH, any kind of thing will be there. So these are some represent things which are put here. So the precursor could be some sugar phosphate. So it could be the glucose 6 phosphate or the fructose 6 phosphate, then phosphoenol pyruvate, pyruvate, acetyl CoA, alpha ketoglutarate, succinyl CoA, oxaloacetic acid, etc. So in all, there are some typical 12 precursors which has been identified, which are generated through the catabolic reactions when the carbohydrate it enters inside the cell. Now these precursors, along with the energy which is being produced in total, in collaboration, they can be utilized for the biosynthetic reactions. So anabolic reactions which are fine. So the synthesis part will take place, then this synthesis will lead to some building blocks, and these building blocks can be then further again organized, and you may get some specialty products like which I told you, it could be the pili, it could be the flagella, etc., etc. So all those particular type of the specialty products can be formed, or you may get these precursors which are then involved into the growth of the organism, means um, it can be responsible for the formation of the different type of the compartments or the organelles which are required for the overall growth of that particular organism. Fine. So this is what we have already studied, or we are aware of this. This is the these two types of reactions which are very important here, that is catabolism and anabolism, and both of them, they are responsible for the overall growth of the organism, right? So these are the 12 precursor metabolites which has been studied, or which has been found to play an important role in the E. coli system. Okay, now here the data which I have presented is in the mammalian cell. You may have maybe the similar or somewhat different in the yeast, also in the mammalian cell or in the animal cell. So it more or less it is same, or few minor variations could be there. So what are these 12 precursors? So these are nothing but the intermediates which are the part of your glycolysis pathway and the TCA cycle. If you are aware of these names, the abbreviations are given, and these metabolites, if you see here in the last column, it is given that they are responsible for synthesis of the building blocks, okay, which can be further converted into the macromolecules or the organelles or some specific structures, right? So I don't think so that I should read it out everything. Only you can one or two I can just tell you that glucose 6 phosphate, it can be used for glycogen production, or LPS, lipopolysaccharides, if you know. Then you have this glycerol 3 phosphate, glyceraldehyde 3 phosphate, so it could be utilized for the synthesis of some typical amino acids like cysteine, glycine, serine. Then you have the succinyl CoA, which can be utilized for methionine, lysine, some tetrapyrols, that is heme, etc., etc., oxaloacetate, aspargine, aspartate, and the other amino acids and nucleotides. Fine. So these are the typical 12 precursor metabolites which are basically converted into these building blocks, and these building blocks can be then converted into the macromolecules, and those macromolecules again they get organized to form the specialized structures. Now if you have these precursors or these metabolites which are being synthesized, so obviously for their synthesis you require the energy. So scientists or the researchers, they have worked upon it, and they have tried to calculate that how much amount of energy is required to have the building blocks or for the synthesis of the macromolecules or the structures. So some example, if you see here, that for the production of the protein, DNA or the RNA, the typical things what we have, we require huge amount of ATP. Like for synthesis of the protein, around you require 39.1 millimolar of ATP per gram of protein. If you want the entire protein to be synthesized in the cell, you require around 39.1 millimolar of the ATP. The same way for RNA, per gram of RNA you require 7.4 millimolar. For per gram of DNA you require 11 millimolar of ATP. Okay, so this gives us an idea that how much amount of energy which is being actually produced by the cell is utilized for the synthesis of the different type of the building blocks. Fine. So here, see, I am talking only about the biosynthesis part of the building blocks. Now there are many building blocks which we are aware of. So that energy which is being produced, it is bifurcated, it is divided into so many what you can say branches now. Okay, so one part of the energy which you can think that it is going for this biosynthesis, some part it is going for the maintenance, some part for the signaling, some part for the secretion, some part for the excretion, etc., etc. Yeah, so this the production of the biomass, the enzymes, the plasmids, mRNA or the synthetic RNA, they not only consume the carbon building blocks, but they also are the energy molecules. Okay, like PEP, what we have seen. So I told you something about then energy rich compounds. Okay, so that kind of compounds could be formed, which could be responsible for providing the energy. Okay, so majority of the energy which I told you could be utilized for the synthesis. The second large amount of ATP which is being utilized, it is for the maintenance, cell maintenance. And then within the cell maintenance, you may have the different type of processes like energy spilling, mitosis, cell component repair, and resynthesis of the macromolecules, etc., etc. And the third one, what you can say, that is the actual production of the biofuel. In this case, if you are going for some biofuel, so for the synthesis of the biosteel molecules, you may require the ATP or the NADPH. Right now, for example, you can take the fatty acid production. So for the synthesis of a particular fatty acid, you may require example इथं जे दिलंय palmitate, that is 16 carbon fatty acid. If you want to synthesize, you require around 7 ATP for the molecules, and you require the carbon skeleton in the form of acetyl CoA. So all this energy and the carbon compounds that together form the biofuel. So this slide will give you an idea about the bifurcation of the total energy which is being produced inside. Now, knowing this, we can now go for the carbon yield, that how much amount of biomass is formed, and whether the energy which is actually produced, whether it is being efficiently used or not. So we'll talk about carbon yield and the energy efficiency. So if you are talking about the central path, which we have discussed, so theoretically around there are 38 ATP molecules which are produced when one glucose molecule is being oxidized. Right? So I think you have already understood the glycolysis and TCA cycle up to the electron transport. So glycolysis and TCA pathway, they contribute to 4 ATP, and different from the oxidative phosphorylation, where we are considering that NADH is producing 3 ATP and FADH2 is producing 2 ATP. Right. So with these assumptions, we have calculated that after the oxidation of one glucose molecule, you are getting around 38 ATP molecules. Okay. Now this process of forming ATP from the ETC, as we know, it is called as oxidative phosphorylation, and in that I told you that there is one factor which is being calculated, that is called by P by O ratio. Okay, So what is this P by O ratio, which I told you that it indicates the number of phosphate bonds for each oxygen atom used as an electron acceptor. Fine. NADH, what we have considered here is the 3 ATP phosphates, while for FADH2 it is 2 P by O ratio. Okay. Now here this was in the aerobic condition, but what will happen into the anaerobic condition, where we have seen that under anaerobic condition the pyruvate is converted into what? The alcohol. Fine. So your pyruvate will not be converted into acetyl CoA. It will not undergo the oxidative phosphorylation. So the energy yield here, it is what? Decreasing. Okay, you are not getting that sufficient amount of energy in case of the anaerobic condition. But for the functioning of the cell, it requires the energy. So in that case, what happens? Okay, so in that case, many a times the acetate is being generated. Okay, so if you remember, when I was talking about the fermentative pathways, I have shown you one separate path where your acetaldehyde can be converted into acetate. Acetate will be then converted into the acetyl CoA, where it can undergo the oxidative phosphorylation and the energy can be produced. Fine. So that shortage of energy could be what you can say, covered up by the formation of the acetate and then the acetyl CoA, so that the energy can be obtained. Fine. So whatever may be the condition, cell is going to accommodate to that condition. Okay, and while it is accommodating, it will try to fulfill the energy requirement which is required for the overall functioning and the maintenance of the cell. Okay, so by hook or crook, you can say here that the cell is producing the sufficient amount of energy which is actually required for its growth and development, whatever may be the condition. Okay. Hmm. So typically, what you can say, for a stable organism which is growing at standard conditions, you can say that it will have a specified P by O ratio of 0.5. But if you are engineering cell, okay, if you are going for metabolic engineering, where you have modified the cell, so in that modified condition, what is going to happen? That majority of your energy which is produced, that will be converted on the basis of the product in which you are interested. लक्षात घ्या मी काय सांगायचा प्रयत्न करतो. हे बघा, नॉर्मल सेल असेल त्याचा एक टिपिकल पी बाय ओ रेशो हा ठरलेला असतो. सो इट विल इज समथिंग नियर बाय टू 3 P by O ratio. But if you have modified a cell, okay, means the modification could be through overexpression of some genes, it could be the suppression of some genes, some kind of modification you have done which is responsible for production of a huge amount of proteins, whereby the reaction in which you are interested is carried out. So what is happening here, that the total energy what the cell is producing, out of the total energy the cell is producing, majority of it will be diverted to the production of the compound in which you are interested. Now when the energy is being diverted toward the production of that compound, okay, so here you can say that energy which is actually required for the growth and the maintenance, it will decrease. Okay, so those particular type of the microbial system or the host, they will have a very poor respiration capability. So in that case, the P by O ratio, it could decrease to 1.3. And if the P by O ratio is decreasing, your cell is not functioning properly. Okay, and maybe eventually the cell may undergo the death. So if you are going to do any kind of modification, you should see that the respiration efficiency of the organism is also maintained. I think you got the point what I want to say. So in this case, what are the things which can happen? So if the host suffer from the severe ATP limitations, की majority of your जे तुमचा ATP produce होतोय तो तुमच्या बायोफ्युएल प्रोडक्शन कडे जातोय. ओके, मग तुम्ही काय म्हणाल सर, मग आपण त्यांना खूप जास्त खायला देऊ. So even in that case, if you are providing the cells with a very good nutrient, still it is not going to affect, okay, or it is not going to regain the strength of the cell so that it can grow for a longer. So it is something like that, की तुमचं जेवण झालंय आणि तरी तुमच्या समोर तुमची आवडती मिठाई आणून ठेवलेली, तुम्ही खाऊ शकाल का? तुम्ही म्हणा हो सर, तरी तुम्ही थोडंफार खाल. But still you come to a level, okay, that where even if you like it, amount of the food in front of you, you are not going to eat it. So it is the same thing like that. So if the host is suffering from severe ATP limitations where the respiration efficiency has decreased, so in that case, even if you are providing and the overall process is going to be an futile process, right? So it is of no use. Okay, and that is why the metabolic engineering approaches are usually carried out into those particular strains which have low product where which are of the category that is the low productivity strains are there, okay, which do not require what you can say a proper p bio ratio of 3 or something like that. Okay, so there is a need that whenever you are going for modification, the priorities toward high carbon yield and energy efficiency have to be carefully balanced during the strain development of for bioproduction. Fine. So you go for the modification but you also try to see that whether the respiration efficiency is being maintained or not, or it is balanced. Okay, so it should not happen that the overall all amount of energy which is being produced production only. It should be also it should also have some sufficient amount of energy for its growth and development, right? So yeah, now we'll talk about how much amount of energy is actually produced, and out of that utilized for the production of the energy sources, that is the ATP or the NADPH, right? The oxidation of glucose in the cells, it has been found that when one molecule of glucose molecule is undergoing this process of oxidation, around minus 686 kilocalorie per mole. Energy is never negative. The negative side indicate that the energy is produced. Okay, so it is 686 kilocalorie amount of energy is come. That is it that the cell is converting this whole energy into the usable form of energy, that is the ATP. It whether the efficiency is 100% or something else. So you can see here that when your glucose molecule is entering in the cell, it is undergoing the so in glycolysis it is forming the two pyruvate molecules. Those two pyruvate molecules will be oxidized to acetyl CoA. So up to this particular point, if you calculate that how many produce, so you can see here there are 4 NADH, so 12 ATPs and 2 ATPs which are directly produced. So and hydrolysis of 1 ATP is giving you 7.3 kilocalories of energy, minus 7.3. एका ATP पासून minus 7.3 मिळतं. So how many? Here it is 2. So 12 into 7.3, 2 into 7.3. If you do the calculation, so up to this point, that is up to the production of acetyl CoA, there are 14 ATP and you are getting 102.2 kilocalories per mole. Okay, this much amount of energy is being produced. Now further, if you go into the TCA cycle and the oxidative phosphorylation, what actually happens? Okay, so from one glucose molecule, you are getting two pyruvate, two acetyl CoA. Now these two acetyl CoA are entering into the TCA cycle. So when it undergoes the TCA cycle, we have the NADH and the FADH2 which are produced. We are also considering the GTP which is produced. So if you see here, it is being calculated and put. So from 6 NADH you are getting 18 ATP. From 2 FADH2 you are getting 4 ATP, and those 2 GTP you can consider them as the 2 ATP. So if you do the calculation, the total is 24 ATP molecules are formed from the two acetyl CoAs which are entering into the TCA cycle. So how much energy you are getting? 175.2 kilocalories per mole. Now if you add these ATPs and if you add this energy, what you get? So in total you get 38 ATP molecules which are generated. So 38 into 7.3, that is the energy produced by 1 ATP. So you are getting how much? 277.4 kilocalories per mole. So what does it indicate? See, when one glucose molecule is oxidized, how much energy is released? It is 686. So out of this 686 kilocalories which is being produced, only 277 kilocalorie of the energy is being utilized for the production of the ATP. So from this you can calculate the efficiency. So the efficiency can be calculated that 100% terms मध्ये जर बोलायचं म्हटलं, so it is 100 into 277 divided by 686. So you are getting only 40.4% of means out of the total energy which is produced after the oxidation of one glucose molecule, only 40% of the energy is being utilized for the synthesis of ATP molecule. Okay, and this amount of energy which is being utilized, they have tried to compare it with the steam power plant. So they found that it is more or less same. But then what happens to the remaining energy? हे बघा 686 पैकी फक्त 277 एनर्जी जर वापरली जाते, what happens to the remaining energy? So that remaining energy, it is simply dissipated as the heat. Okay, and that heat is utilized for the maintenance. With for maintaining the body temperature. Okay, and it could be used for some other processes also, where we don't want to go in all those particular type of the details. Okay, so here the energy efficiency is simply only 40%. So the approaches has to be made whereby you can maximally utilize this energy which is being produced. But if you're going for it, say you if you try to increase this efficiency, means from 40% to 60 or 70%. So by doing this, what you're doing? You are not providing sufficient amount of energy which is actually utilized for the maintenance of the cell. And if the maintenance is not taking place, obviously it is going to affect the functioning of the cell. Okay, and the cell may undergo the process of death. Okay, so these things you have to take into consideration if you are going for any kind of modification into the system. Okay, what I have tried to explain. Yes, am I audible? Hello.
Yes, sir.
Okay, so this was about the carbon yield and the energy efficiency. Yeah, this one figure which I got, which will give an idea about the energy efficiency. So if you see, this is a microbial system. One cell has been shown. So on this side, the different substrates which can be used. So you can have any one of these kind of substrates. So whatever is the substrate when it enters, okay, inside the cell, it will be catabolized by the typical center utilized for the production of the different type of the precursors. Energy will be produced. Those precursors and energy can be utilized for the biosynthetic process, etc., etc. Now if you see here that if you are modifying this system, so what is happening, that the substrate which is undergoing the central path, instead of that the substrate is diverted toward the production of the compound in which you are interested. Now by doing this, what is going to happen? you are going to prepare a system which is having a very low P by O ratio, where the ATP maintenance or the say maintenance of the cell is also not proper, and you are going to have a futile cycles. Fine. So this is one of the research article which is published into the Frontiers in Microbiology, which is trying to explain the things which I explained in the previous 3 4 slide about the carbon yield and the energy efficiency. Right. So maybe I may not go in that details of this, but still I will explain the different types of the products which could be operated. Right. So leave it. So I think the idea is clear. Now we'll try how to go for modeling, and that too for mathematical type of the modeling. थोडक्यात बघू आपण. I'm not taking you into the complete details, but I'm just trying to give you the overview or the basics that how you can go for. Action. So according to that, just 1 minute. Yeah, so this law of mass action, it was given in 1864 by Waj and Guldberg. So this so this states that the reaction rate is proportional to the probability of a collision of the reactors. Collisions has to occur. There has to be the collisions between the reactants. Okay, and depending upon that, at how much time these molecules, sorry, these substrates, that or the reactants, you can say here that how much time these reactants are taking to come closer and have collisions between them so that the reaction can occur. It can be predicted by the law of mass of action. Okay, so the reaction rate is proportional to what? The probability of a collision of the reactants. Okay, and this probability in turn is going to depend upon the concentration. Right, but practically the concentration cannot be calculated, so we usually talk in terms of molecularity, that how many molecules are present. Fine. So how many molecules are actually taking part into the in the reaction, that is going to decide the rate of the reaction. Fine. So we consider this equation or this reaction, that is A plus B is converted into C plus D. This small j, k, l, m, these are the concentration which are being denoted here. So as per the law of mass of action, the K equilibrium, okay, so the equilibrium constant can be denoted that ratio of the product by the substrate. So it could be put like this: concentration of the product, that actual product, and the number of molecules which are mentioned here, so which are the superscript here. So it can be written like this. There is an another way to express this, what you can say, the rate of reaction, and that is the ordinary differential equations. That is called as what? Ordinary differential equation. Okay, so where this ODE, it helps us to understand the change into the concentration of your reactants or the products, the reactants which are utilized and the products which are formed. So that uh, what you can say, that dynamic thing happening for the reactants and the product can be obtained from this ordinary differential equation. So for this, here we calculate the derivative with respect to the time. Okay, so take an example that S1 plus S2 is converted into 2P, and 2P can be again converted back to S1 and S2. So these things, they can be expressed in the form of derivative with respect to the time. So it can be written as d by dt into S1, which could be equal to d by dt of S2, which is equal to minus V. Okay, as these reactants, their concentration is going to decrease. So always if it is decreased, so we represent it by minus V. So minus V is nothing but the rate of reaction here. Fine. Or if it is a reversible reaction, it could be denoted as d by dt into p of p, derivative of p with respect to time, which is equal to 2v, because there are two products which are being utilized. Fine. So these are the two ways where you can express the rate of the reaction, or where you will be able to express the concentration of your reactants and the products which are taking part into the reaction, the actual reaction. Okay, so these are the two ways where you can go for it. Now consider this system where the reactions are taking place, that there is something here which is converted into S1, S1 is converted into S2. So there are two S2 molecules which are generated. Further, two S2 can be converted into something else. Then S1, it has an another fate, that it can be converted into S3. Okay. So now each and every reaction which is occurring into this system, it will have its own rate of occurrence. So we can denote it by V1, V2, V3, and V4. Okay, so whatever may be the complexity of your system is there, that there could be thousands of reactions which are involved into this. Each and every reaction has its own rate constant. Okay, which is denoted by V1, V2, V3, or V4. Now this what you can say, the metabolites which are involved and the rate of their reactions, they can be represented in the stoichiometric form. Okay, so this is very important, that is the stoichiometric matrix. So you should be able to represent these reactions, or these reactions in some typical format, and that is nothing but the stoichiometric matrix, which is denoted by capital S. So it can be represented like this. So this is what is the information which we have converted about these reactions in the form of numbers. So it could be 0, 1, -1, etc., etc. Okay. Now how to read this matrix? Now these columns, what you can see, these columns, they are denoting the reactions, that how many reactions are there into the system what you are trying to study. Now here there are four reactions what we are doing, that is V1, V2, V3, and V4. So these are the four reactions what you can represent. So each column is representing the four reactions. Now these rows, they relate to the number of metabolites which are involved. Now how many metabolites can we see here? It is 1, 2, and 3. So there are three metabolites. So okay, so here we are trying to see the participation of this metabolite into these four different type of the reactions, and they are represented in the form of these numbers. The same thing for your S2, the same thing for your S3. Okay, and from this you can calculate the d by dt, that is the derivative with respect to the time for each and every metabolite. Now what does this 0, 1, -1 indicate? 0, it indicates that this metabolite is not playing any kind of role into that particular reaction. 0 का indicate करता है that it is not involved into the reaction. 1 is indicating what? That it is a reactant. Sorry, 1 is indicating that it is a product which is going to form. म्हणून positive असतो तो. And -1, it is indicating what? That it is a reactant, because reactant is going to be utilized, and that is why we represent it by minus. Okay, तर ह्या काही बेसिक गोष्टी आहे, फक्त तेवढ्याच आपल्याला लक्षात ठेवायचं आहे. As I told you, we are not going in all the details of this. So this is how you can present your reactions of a particular pathway in which you are interested in the form of stoichiometric matrix. Fine, like S1. Now S1 is formed by this V1. So V1 मध्ये आपण 1 लिहिलंय. आता S2 मध्ये होतोय, मीन्स इट इज बिंग युटिलाइज, म्हणून -1. V3 ची रिएक्शन घेतली, तिथे S1 चा काहीच रोल नाहीये, सो इट इज 0. S1 is again it could be form or it could be utilized. into S3. So V4 we have presented it by 1. जर आता S2 चा विचार केला आपण, we talk about S2. So S2 V1 मध्ये participate करते का ते? नाही. There is no direct correlation. Then what about V2? V2 मध्ये yes, it is being produced from S1. So there are two molecules. So it is represented. V3 means it is converted into something, means it is being utilized. So -1. V4 मध्ये it is not taking part. So 0. Same thing for your S3. S3 is not taking part in V1, V2, V3. So all these three we are 0. Only thing is that it is produced from this S1. So you have only it as a 1. So this is how to read this matrix, or how to represent your reactions into the form of a matrix, that is stoichiometric matrix. So from this you can calculate the d by dt for S1, which is V1 minus V2 plus V4. So V1 पासून तो तयार होतोय, त्याचं कन्वर्जन S2 मध्ये होतोय, means it is being utilized. So minus V2 plus V4, because it is also converted into S3. म्हणून V2 plus V4, which is being negative, it could be minus. The same thing for d by d2 for S2. त्याचं काय असेल? 2V2 plus V3. Then for d by dt for S3, it is V4. Right? So this is how you can represent your reaction pathway in which you are interested in the form of an matrix. Now you take an example, then actual example here. Suppose this is a cell. A substrate is entering into the cell. It is first converted into A is converted to B, A is can be converted into C, B will be converted into product 1, C is converted into product 2. So I have shown only these 5 reactions. So each reaction, as I told you, it could be denoted by its rate constants, or the rate of reaction. It will have a typical rate of reaction which is denoted by V1, V2, V3, V4, V5. Right? So here, if we talk about, there are some reactions which we can consider. The first one is that substrate to A, which is denoted by V1, A to B, V2, A to C, V3, B to product 1, V4, C to product 2, V5. Now, if you really have understood the matrix in the previous slide, you should be able to create a matrix for this reactions, these 5 reactions. So as an assignment, you can now try to create a matrix. Okay, so maybe in some other presentation, we'll come back to this example and we'll try to prepare the matrix for it. And from this, you can calculate the reactions, or the equations for this, that is dA by dt, dB by dt, and dC by dt. So take it as an assignment, try to solve the matrix if it is possible. Fine. So I think now this is clear, that how to represent it in the form of an matrix. Okay, so maybe I'll stop here, because I have some meeting which is actually going on. I have to go there, and already it's an up to 11 o'clock. So I think so I will stop here. Any difficulty up to this point? Yeah, any questions, any doubts? Yes, put your roll numbers in the chat box. Sanskruti, send me the numbers.
Okay.
काही डिफिकल्टी आहे का आतापर्यंत?